start. You are recording. Yep. So recording works, and I share the screen with you, which hopefully you should be able to see the screen. Can you see the screen? Wonderful. Oh, I love that. We get some background music. Actually, I was thinking it's really a good thing that you have this list of people on the so-called clown list, because if somebody forgets to record their or register their attendance, we can always look at the clown list, can't we? Absolutely, I fully agree. But Kenneth, that's not something that you have to that you have to take up with me. This is something with your fellow course mates. Well, we discussed that yesterday. Don't drink water. Fish do unspeakable things in it. I better not look too close on that. What happened? Ah, there are weird things going on. Can you still see it? Can you still hear it? Can you still see the screen? Yes, because I get messages that the meeting has ended also, but that's not true. Good. So everybody has registered attendance. That's great. And as I said, I think you all are pretty knackered. So that's the reason actually why I'm not giving you a lot of asynchronous activities, because I have a pretty good idea that, you deserve a little bit of a break. Am I right or am I right? Ask me, Daria, ask me. No, I'm going to have something to eat. Oh, it's the dreaded book question. I would say buy the book that is right for you. Check out in the library, check different books, and see what is the best book that helps you learn most efficiently. I think that's the best advice that I can give. And do the test. The more you agonize over it, the more you agonize over it, the more stressed you get. And I can tell you, it is so easy. I showed it to my kids, and they say, "Well, really, is that what university is about?" So please, please, please do the test. Okay, I think we can probably make a start. No, they are not really the kids of a scientist. Although my son has decided that he is going to do for his A levels, he's doing maths, chemistry, and biology. I have no idea what possessed him, but hey. Anyway, let's get started for the last act of this week. BI 300, Introduction to Biochemistry. And what I want to discuss with you today is the genetic code and amino acids. And just to get started with, well, don't forget to register your attendance in Moodle. Very important. And I talked about that. What I'm going to tell you is you do not need to learn the genetic code by heart. That is total waste of brain power. And yes, you need to learn amino acids, but I don't ask you to draw the structures, but you have to recognize the amino acids and you have to know about their properties. So please, no genetic code, yes, amino acids. Okay, so what we have done so far, we've talked about DNA, and let's just quickly revise the sort of the central dogma. DNA, the DNA can replicate, and I indicate that like that. Learn the names and stuff for the amino acids, absolutely. And the way you revise is really up to you. You need to find what's the best way for you. I think that's really important. And I want to point out what we are doing here. All the topics, at least for the stuff that I teach, there is so much material out there available on YouTube and God knows on blog posts. You find so much stuff out there. So if you get stuck on a topic, just go through the world's second largest search engine. Do you know what the world's second largest search engine is, actually? YouTube, absolutely right. It's YouTube. It's the world's second largest search engine. That's YouTube. So we have DNA. DNA is converted into messenger RNA. And this process is the transcription. The messenger RNA then, with the help of ribosomes, is then converted into proteins. And this process is called translation. Translation. And the proteins will do weird and wonderful things. For example, they fold into their correct structures. And we will discuss that quite a bit. So what do we need in order to make proteins? We need to have amino acids. Amino acids. And the right sequence for the proteins that we make is encoded in the DNA and in the mRNA. We have about 20 different amino acids. Some people say it's 21. So let's quickly do a calculation what our DNA needs to look like. So we know that the DNA, when we are looking at a gene is made up in the DNA of codons, and each codon is a triplet of bases. Triplet of bases. Let's do a little bit of probability for that. So we said when we have a triplet, we have 4 bases. So with one base in a codon, one base, how many different amino acids could we encode? So we have the position A, T, C, G. So in theory, we could do if we just had one position, we could get 4 amino acids and abbreviate amino acids with AA. Okay, if we had 2 places, 2 bases, or 2 positions, let's say 2 positions, how many amino acids could we encode with 4 bases? Yep, that would be 16. So that would be 4 times 4. But it would not be enough for the 20 different amino acids. So that would be 16. So 2 positions is not enough. So what do we get with 3 positions? We get 4 times 4 times 4. So that gives us 64 potential amino acids. Amino acids. If we were living on a planet where we only had, let's say, 12 different amino acids, then codons with 2 bases would be absolutely fine. 20 amino acids that we have, and somebody made the right 64 Sulane. Yes, 64. So what was the name of the elephant in 64 Sulane? Does anyone remember? No. Nelson the elephant. Yes, Nelson the elephant. And the little girl was Lucy, wasn't it? God. So And somebody is open here. Georgina the giraffe, yes. Okay, can you please mute yourself, whoever is there? Or you have to sing the song for 64 Sulaim. Anyway, and somebody made a good point. We have 20 amino acids, and we need also to indicate where the gene should stop, where the protein should stop. So we need at least one stop. So with 64 amino, with 3 positions, we can potentially code for 64 amino acids. But since we have only 20 plus stops, we have 2 options. Nature can do 2 things. It might not use all the codons. That's one thing. Or it could use several combinations of the bases for different amino acids. And that is what is sort of named as the genetic code. Genetic code is what is called, what's the word? Genetic code is anyone? Degenerate. Degenerate in this case means that several codons, several triplets, different triplets, can potentially code for the same amino acid. So there are only 20 natural occurring amino acids, but we have space to code for a max of 63 amino acids plus a stop. But we don't use all the 63. We only have 20 naturally occurring amino acids. Some people say it's 21, but I'm not entirely sure. And degenerate because there are triplets, different codons, that code for the same amino acid. So a lot of people say there is redundancy. Why 21 then? There is an additional amino acid that occurs in some proteins, which is not included in the genetic code, which is made in a slightly different way. I'm not going to touch on that. And we need 20 amino acids and an indication where the protein should stop. So that's why we have stops. So that's what is called, the code is degenerate. And different triplets can code for the same amino acid. And people usually say, well, actually, it doesn't really matter what triplet I use as long as it's coding for the same amino acid. And I will demonstrate to you that this is wrong. The code is not degenerate. And there is a purpose behind the different triplets that we have. What does it look like? Look at my beautiful wheel. Yeah, you've been lied to. You have been lied to. The genetic code is not degenerate. Somebody's asking, why is it not universal? I come back to that question in a minute. I think, yes, you can. You can use codon and triplets interchangeably. I tell you, at the right time, I will tell you. But you are not yet ready for the truth, Kenneth. So how does it work? Here is a wheel for the genetic code, and it tells you which codon, which triplet actually codes for an amino acid. So for example, we start from the inside and go outside. So the first codon, the first base, if you say is a U, uracil. The second one, again, let's put a U there. U, that's the second. And the third one, let's pick this C here. So the triplet UUC would stand for the amino acid PHE, Phe, which stands for phenylalanine. And what you will need to learn is the name of the amino acid, the 3-letter code, that's this Phe, and the 1-letter code. And in the case of phenylalanine, the 1-letter code is F. And I will give you a list. And you don't need to learn what codon or what base combination gives you the amino acid. So you don't need to learn the code, but you need to know how to apply it. Okay? You have to learn all 20. You have to learn the amino acids. So here's a question for you. Using this wheel, which amino acid is encoded by CGA? Just give me the, yep, it's ARG. It's ARG. This is the amino acid arginine. And the one, and how did you do that? So CGA, that gives this ARG here, right? Does that make sense? It's pretty straightforward and easy. And the 1-letter code for arginine is an R. Will you get the wheel in the exam? If I ask you a question like that, then absolutely yes. Right, now you have been told that the genetic code is universal. Every single organism on this planet, yes, you need to know the 3-letter code, you need to know the full name, and you need to know the 1-letter code. But there are plenty of mnemonics out there. And my suggestion is the more outrageous a mnemonic is, the better you can remember it. You have to learn the 1-letter code. Very often it's quite easy, but sometimes, it's not absolutely intuitive. So you've learned that, oh, you've got a good brain. No, you don't need to memorize the different codons. You just need to remember what the amino acids are, what they are called, and how they are abbreviated. So don't stress about the code. Yeah. So you learned it is universal. Every single organism on this planet is using the same code. And I have to tell you the truth. I have to be honest with you, it's not true. I will give you don't worry about the one-letter codes. It is not true. You have been lied to. It's not universal. And unfortunately, I have been at the receiving end of that because I did my master's in the Institute of Genetics. And my job was to take a gene from yeast mitochondria, put this gene into E. coli, and we use certain DNA transport mechanisms, plasmids, into E. coli, and in E. coli produce the mitochondrial protein, and then study it. So that was the aim of the project. So I did. And we knew that the yeast mitochondrial protein was about 300 amino acids long. That was the mitochondrial protein. And what we got was always about 230 amino acid in E. coli. And we could not figure out what was wrong about that. Why did we always get a shorter protein in E. coli? In the mitochondria, the protein was nicely produced. In E. coli, when we tried to produce it, it always gave us a shorter protein. When we took other proteins from other organisms, like insulin or something, we got the right size in E. coli. So in E. coli, there wasn't anything wrong with it. Yeah, I know bacteria doesn't have mitochondria. We took the yeast mitochondrial gene and put that into E. coli. And that worked perfectly fine. It worked with other yeast genes. So the yeast genes in E. coli usually were exactly the right size. So what was going on? And I was really desperate. And I remember about 35 years ago, I was sitting in the tea bar. I was blinding and effing about this damn mitochondrial protein. And then one of my coworkers said, wait a moment, yeast mitochondria, they use a different codon, don't they, for the stop codon? And I was, yeah, George, can you please pass the Wait, what did you just say? What the heck? And it turns out that yeast mitochondria use the codon UGA. They use this UGA for the amino acid trip, which is tryptophan. The one-letter code is a W for tryptophan. What is UGA in all other self-respecting organisms? It's TER, Look at that, ter. Do you know what ter stands for? Ter stands for termination. It's a stop codon. So in mitochondria, the mitochondria, when they produce the protein, they were quite happy. They put the tryptophan in. But when we used the gene in E. coli read the UGA codon as a stop codon and therefore always gave us a smaller protein. Life sucks sometimes, right? So what, and I lost basically 2 months of my project. Once we figured that out, that the code is not universal, it wasn't a big problem. We just simply modified this TGA, and you can easily do that. We modified it so that it is read as a tryptophan in E. coli, and we got the right protein. Yep, that is what happened. So what on earth is an amino acid? Well, a typical amino acid looks like this. We start with a carbon, which has hydrogen. It also has an COH, and it has this group. And I deliberately draw it like that with an R. This R just simply stands for rest. And the amino acids it's mostly universal, but not always. And Sod's law, sometimes you run into that without knowing. I got burned. I always check when I work with a weird organism what codons they use. So we've got different groups here. So what is, oh, let's make it a little bit colorful. What is this group here known as? COOH. What is it? It's a carboxylic acid. Righty-ho. Carboxylic acid. What is this group known as? That's an amine. So what we have is an amino acid. That is what an amino acid looks like. This carbon here that we have, the piggy in the middle, that is also called the alpha carbon. And this has a particular property. This alpha carbon for most amino acids is chiral. It is the central carbon. It is the alpha carbon. It is chiral. What does that mean? When you stand here in the carbon, in the middle, and you look up to the left, down to the right, this carbon will always see four, or in most cases, it will see four different residues. This means that it cannot be superimposed with its mirror image. We know that everything in life has a mirror image, with one exception. Is there one thing that doesn't have a mirror image? It is a stereoisomer, absolutely. It is stereoisomer, it's asymmetrical, and if I draw the mirror image structure, you can't superimpose them. And it's also a tetrahedral structure. You can't superimpose them. Is there anything that does not have a mirror image? And that's exactly, yes, you can't fit your hand over the other exactly in the same way. That's what chiral means. Chiral comes from the from the Greek word kairos, which means hand. And a characteristic for a chiral compound, for a stereoisomer, is that it has four different groups attached. So here the amino acid, hydrogen, rest, and the amino group. Which thing doesn't have a mirror image? Does anyone know? No? Yeah, glycine doesn't have a mirror image, but for a different reason. Anyone else? What doesn't have a mirror image in life? Vampires, exactly. Vampires don't have mirror images. So that's the easiest way to find out whether your partner is a vampire. You just put them in front of a mirror. And if you can't see them, you either need to go to Specsavers or it is a vampire. Right. So we are always dealing with amino acids that are in this side of the mirror, where the amino group is pointing to the left. And this is an L amino acid, whereas this here would be a typical case of a D amino acid, where the amino group is pointing to the right, which is dextrose in Latin. Dextro in Latin. That's why it's D. I'm not getting sucked into this discussion here. So we always are dealing with L amino acids. It does not mean that D amino acids do not exist. Some bacteria can actually produce D amino acids, but it's not something that we are going to discuss. So here comes the guide to the 20 common amino acids. And I'm terribly sorry. This is the stuff that you have to learn. I'm not asking you to draw the structure. But I need you to know, if I show you, for example, an amino acid. So, for example, I show you H2NCH. COOH. This is the easiest amino acid. This is actually the amino acid glycine. GLY. And the one letter code is G. So the L, in the L form, the amino group points to the left. And in the D form, the amino group points to the right. That's the difference between L and D. So we have glycine, Gly, and G. Yes, you need to memorize all 20 and their properties. I know. Big question, is glycine chiral? No, you totally nailed it, because glycine has two of the same groups, the hydrogens here. So glycine is the only amino acid that is not chiral, because it doesn't have in this alpha position the same groups. By the way, we always write amino acids so that the amino group is at the left and the acid group is to the right. And we write all proteins so that the amino group is to the left and the carboxyl group is to the right. Okay. So how do we make a longer peptide? So a peptide is everything that contains two or more amino acids up to a length of, let's say, up to, say, 10 amino acids put together. And we use what is called a condensation. That's absolutely right. H, N, H, C, A, C, O, O, H. Let's do the next easy amino acid. Let's put a methyl group here. This is the amino acid alanine, abbreviated ALA or A. And a condensation reaction is basically we combine the OH group and the hydrogen from the nitrogen. We remove water. Oh, let me draw it like that. We remove water from it and we get H, N, H, C, H. That was our glycine, CO, N, H, C, H, CH, 3, CO, O, H. And this is what is called a peptide bond. So let me draw this peptide bond out again. Slightly larger. This here is called the peptide bond. Chemically, what would this bond be called? It's not a glycosidic bond. It's not a glycosidic bond because we don't have any sugar in it. It's amuse. I'm very amused. It's an amide. Exactly. It's an amide bond, which is basically a combination of acid and amine, an amide. And an amide bond, this peptide bond, has another really interesting property. If you have a normal bog standard bond, let's say a CC bond, then everything can rotate around this bond. It sort of, can flip around. It can rotate around this bond. It is a covalent bond, yes, and it can flip around. It is equivalent to an ester bond, but an ester bond would have an oxygen instead of the nitrogen sitting there. Absolutely right. So an ester would be with an O instead of the nitrogen. Amide is with an N. So the question is, can you also rotate around this bond here? So could you actually do something like that? NHC. So we just rotate it around. Looks like it, yes. You could think that you could rotate around. But actually, here comes the weird stuff. This electron can flip in here. This one opens up and joins the other one. So what you end up with is something like that. Negative charge, double bond, N, H, C. Nitrogen is positively charged, the oxygen is negatively charged, and now you have a double bond here. We are not discussing the mechanisms. This double bond you cannot rotate around. Double bonds cannot rotate. So this double bond, and actually it's not a total double bond, it does do it naturally, and it's more stable than just this form here. So what people usually show is they show something like that, which looks weird, but it indicates that we've got a sort of a weird system where we cannot rotate around the amide bond. And that means the peptide bond is quite rigid. So peptide bond, the amide bond, peptide bond is rigid. It cannot easily flip around. Does that make sense? At least you learned something. We need to bear that in mind when we then look at peptide bonds and when we look at proteins. Now, we have seen that in a normal peptide bond, and I just write it like this, in the open form, there is no difference between amide and peptide bond. Or if you like, a peptide bond is a special case of an amide bond. An amide bond is just simply CON bond, like I've drawn here. And the peptide bond is just simply this amide that combines two different amino acids or two amino acids. So what we have, again, is we potentially have a hydrogen and we have got a nitrogen here. So what we learn in one of the next sessions is that we can easily form this interaction NHO. Can anyone remember what this interaction would be called? What if we have NH and sort of O? Absolutely right. Yes. Don't put a question mark behind that. You are absolutely right. This is a hydrogen bond. Hydrogen bond. And we have loads of hydrogen bonds in proteins because we've got these NH and the COs in it. One last thing that I need to discuss with you is I have drawn the amino acids like this. H, here is the carbon. Let's go for alanine again, CH3, because that's easy to draw. COOH. And here would then be the next peptide bond or the free amino acid. So, but usually when we are working in solution, we would not necessarily find it like that. We would find actually that this amino group is protonated and that this hydrogen has been released. So what we would find is a structure that looks like HNHH. C, we've got an H here and a methyl group C3 here, and then we've got CO. What is the charge on the O here? What is the charge on the oxygen? Ah, yeah, people are coming up with the right name. So that's the negative charge here. And we would have a positive charge here at the nitrogen. And because we have two charges for this thing, sort of when it can't decide what it wants to be, boy or girl or something like that, this is called the Zwitter ion. Zwitter ion means it has a positive and a negative charge. Well, I mean, yeah, it is both. The biological term for an individual that has both male and female reproductive organs would be a Zwitter, or the German version Zwitter, where it comes from, which actually comes from the word zwei, which means two. And I think because it has some sort of connotation, I think it is a sort of a bit problematic, the phrase Twitter. In biology, you would usually call an organism hermaphrodite, which is again a combination of male and female reproductive organs. And there are lots of examples where this can happen in nature. For example, when you're working with some kind of worms. So, today we've covered a lot of ground. We covered the genetic code, which you don't need to learn. We've covered amino acids, which you do need to learn. And for example, I will ask you we will discuss that the next time. But I will ask you, for example, name three amino acids where R is positively charged. And the difference between the amino acids is always in this rest group, in this R group here. Where is it? In this R. That is where the differences are. And there are 20 differences. So you have amino acids that are charged. I'll put that up on Moodle. Don't worry about it. Yes, I will put that up on Moodle. Don't stress about it. This will go up. But you find this poster, you find loads and loads of things about the amino acid. So I might ask you, which are the three amino acids that are positively charged? Well, they are R, K, and H, for example. Which amino acid is small and polar? So you would need to know these things. You need to know the structure, but you don't need to draw them. But I want you to recognize them. A couple of years ago, we had a student who was incredibly entrepreneurial. She was also very much into beauty and fashion. Absolutely right. Yes, that's the three amino acids. She was also into beauty and fashion. And at that point, the module was taught by somebody who insisted that the students need to be able to draw the structure of all 20 amino acids. So guess what she did? She painted her nails. So she had all the 20 amino acids on her nails. The nice structures of them. Unfortunately, she was so proud of her artwork that she showed it on Facebook. So you have 2 amino acids per nail. Well, it got detected, but luckily it wasn't a big problem. No, it didn't get detected because she got the advice to use some nail varnish before she goes into the exam. Luckily for her, because she painted them and repainted them because she wanted them to look really nice, by painting them on the nails, she learned them. So here is a trick for you. For exams and for tests, make flashcards, make cheat sheets. Because by writing and producing these cheat sheets, you will learn a lot, and it helps you to memorize if you ever have to do some memorization. Don't smuggle them into exams, because we had students who got kicked out of university for doing that. With that, I think we have done enough. Not worry about that. We have done enough for today, for this week. I'm really looking forward to seeing you to our next session. I think it's Wednesday morning again. We have got one when I talk a little bit more about protein and how proteins can fold into proper structures. Until then, thank you very much for bearing with me. Have a lovely weekend and try to get some rest. I'm not setting you any homework. Take care and be good. Don't do anything I wouldn't do. Bye.